endoplasmic reticulum has folds, no? Same way these Golgi bodies also have folds. Therefore, they are reticular structures and they are present close to the nucleus. Cisternae, that is the folded sac-like structures of the Golgi apparatus, it has a particular function wherein it helps in modifying a number of proteins that is synthesized by the ribosomes. Which part of the Golgi body is responsible for receiving proteins and lipids from the endoplasmic reticulum? It is cis phase. Hello everyone, a warm welcome to the session on class 11th biology for CBSE. I'm Dr. Divya, biology faculty, Vidhyaashram School of Excellence, Mysuru. So in this session, let us learn about the organelle Golgi apparatus under the chapter Cell, the Unit of Life. So we'll have a small discussion on this particular topic and then move on to learning about some of the MCQs that can be framed under this particular topic. So Golgi apparatus, it was first discovered by the scientist Camillo Golgi in the year 1898, and he discovered Golgi apparatus or Golgi bodies in the form of densely stained structures which were in the form of folds, and because it was discovered by Camillo Golgi, the name has been given as Golgi apparatus. And these structures are reticulated or reticular structures just like the endoplasmic reticulum. So how the endoplasmic reticulum has folds, no? Same way these Golgi bodies also have folds. Therefore, they are reticular structures and they are present close to the nucleus. Now, don't get confused here. They don't have any connectivity with that of the nuclear membrane, but they are just located somewhere close to the nucleus. That's it. But the endoplasmic reticulum forms a network which continues from the nuclear membrane. So that is the difference between the endoplasmic reticulum and the Golgi apparatus. And the Golgi apparatus, when you look at the structure, they are made up of many flat disc-shaped sac-like structures, and those disc-shaped sacs are nothing but called as cisternae. And the diameter of each of the cisternae is about 0.5 to 1 micrometer. And these Golgi apparatus or these cisternae, in the Golgi apparatus, the cisternae, that is the plate-like or the sac-like structures, are stacked one above the other. And when you look at it, like when you stack plates one above the other, how it looks, no, exactly like that, the cisternae looks in the Golgi apparatus. And the cisternae, that is the number of cisternae, can vary from one Golgi apparatus to the other. Say for example, some of the Golgi apparatus, if they have 8, some may have 9, some may have 10, like that. So it depends. And the Golgi cisternae, they are concentrically arranged near the nucleus. And when you look at the cisternae, I told you they are somewhat oval-shaped sac-like structures, no? They have two phases. One is the trans phase and the other one is the cis phase. So talking about the trans phase, so the cis phase that we say, it is called as the convex cis or the forming phase. And one more is the maturing phase. So what are the two phases in the Golgi apparatus? The forming phase and the maturing phase. So the forming phase is also called as cis phase and it is convex in nature. And the maturing phase is also called as trans phase and it is concave in nature. So convex, cis, forming phase. Concave, trans, maturing phase. Okay. So the cis and trans phases of the organelle are entirely different, but they are interconnected to one another because they have their own specific functions. So this is the diagram of the Golgi apparatus. It is how it is given in the NCERT textbook, wherein you can see the foldings which form plate-like structures or sac-like structures, and they are called as cisternae. And this cisternae has a convex cis phase which is also called as the forming phase. So, cis phase, F-A-C-E, it is also called as forming phase and it is convex. Next we have the trans phase, which is also called maturing phase, and it is concave. So much you have to remember. So these are the two phases that are present in the cisternae. Next, moving further, let us study about the functions of the Golgi apparatus. So the main functions of the Golgi apparatus is to pack the materials or the contents present inside the cell, that is distributed in the cytoplasm of the cell, and deliver it to the intracellular targets. So intra means within the cell or outside the cell. So it can send the whatever packaged material is there, that is there in the cytoplasm, that will be collected by the Golgi apparatus, and it will be delivered either within the cell itself or to the neighboring cell. So that is one of the main functions of the Golgi apparatus. Next, the Golgi apparatus, since it remains in close association with the organelle endoplasmic reticulum, one of the main reason why the Golgi apparatus has to remain in close organization or in close contact with the endoplasmic reticulum is, and that is why Golgi apparatus is present close to the nucleus, okay? Because endoplasmic reticulum is almost a connectivity of the nuclear membrane of the nucleus itself. So wherever the endoplasmic reticulum is there, near that only Golgi apparatus should be there, because both have a function which is interconnected to one another. They depend on each other for a particular function. So what is that? So I told you Golgi apparatus helps in packaging of material, or it helps in delivering the packaged materials either within the cell or outside of the cell. So here, whatever material is being packaged in the form of vesicles, from the endoplasmic reticulum, it fuses with the cis phase. That is, the endoplasmic reticulum, what it will do is, it forms a connectivity with the Golgi apparatus, wherein the cis phase, that is the forming phase or the cis phase of the Golgi apparatus, fuses with the endoplasmic reticulum. Why it is connected? It is because something related to the transferring of the packaged materials in the form of the endoplasmic reticulum from the Golgi bodies to the endoplasmic reticulum, and from the endoplasmic reticulum to the Golgi bodies. So for that particular reason, it is in close contact with the endoplasmic reticulum. So Golgi apparatus remains in close association with the endoplasmic reticulum, as the whatever materials are needed to be packaged, right? So which actually helps in package of the material? Golgi apparatus. And the materials needed for the packaging of the materials by the Golgi apparatus is present in where? It is present in the endoplasmic reticulum. So the endoplasmic reticulum should deliver these materials required for the packaging to the Golgi apparatus, and that is the reason why the Golgi apparatus remains in close association with the endoplasmic reticulum. Golgi apparatus remains in close association with the endoplasmic reticulum, as the materials to be packaged in the form of vesicles, or the vesicles from the endoplasmic reticulum, will go and fuse with the cis phase of the Golgi apparatus. Once it fuses with the cis phase of the Golgi apparatus, from the cis phase it will move to the maturing phase, that is the trans phase. From the cis phase, it will move to the trans phase. And the cisternae, that is the folded sac-like structures of the Golgi apparatus, it has a particular function wherein it helps in modifying a number of proteins that is synthesized by the ribosomes. So those ribosomes which are present on the rough endoplasmic reticulum, whatever proteins is being synthesized by those ribosomes which are present on the rough endoplasmic reticulum, that will get modified by the cisternae of the Golgi apparatus, and then that modified proteins will be delivered to where? It will be delivered to the trans phase of the Golgi apparatus. So cisternae of the Golgi apparatus helps in modification of a number of proteins which are synthesized by the ribosomes that are present on the rough endoplasmic reticulum before these proteins are being delivered from its trans phase. And the Golgi apparatus is one of the most important sites for the formation of glycoproteins and glycolipids. So these are the functions of the Golgi apparatus. So they help in synthesizing or forming glycoproteins and glycolipids as well. So what are the main functions? One function is to package the materials and deliver it to the respective sites either within the cells or outside of the cell. One more function is it always remains in close association with the endoplasmic reticulum, so that whatever materials are being delivered by the endoplasmic reticulum in the form of vesicles, that gets packaged by the Golgi apparatus, wherein the endoplasmic reticulum will fuse with the cis phase of the Golgi apparatus, and once those packaged materials move from the endoplasmic reticulum to the cis phase of the Golgi apparatus, then it moves to the maturing phase or the trans phase. That is also one of the functions. Next, the cisternae in particular, it helps in modification of the proteins, that is ribosomes on the rough endoplasmic reticulum, and apart from that, the Golgi apparatus also helps in the formation of glycoproteins and glycolipids. So this is about the function. So now we know about this topic. Let's see what all sorts of MCQs can be framed regarding that particular topic. Which organelle is responsible for packaging, moving and sorting proteins? Is it ribosomes? Is it Golgi bodies, endoplasmic reticulum, mitochondria? Now we can eliminate mitochondria because mitochondria doesn't synthesize proteins. The main function of the mitochondria is to produce ATPs. That is why they're called as the powerhouse of the cells. Now the option is between ribosomes, Golgi bodies and endoplasmic reticulum because all the three somewhere they're related to the proteins. Now keeping these three in mind, we have to go back to reading the question again. What is the question? Which organelle is responsible for packaging, modifying and sorting the proteins? Ribosomes, their function is to just produce the proteins. Endoplasmic reticulum's function is whatever ribosomes produce the proteins, they'll package all those materials in the form of vesicles and deliver it to the Golgi bodies, or they deliver these proteins to the Golgi bodies. So now the organ which is particularly responsible for packaging, modifying and then sorting the proteins and sending them to respective sites is done by the Golgi body. So option B is the right answer here. Next question. Golgi bodies are composed of a series of flattened sacs. So what are the flattened sacs called? Are they called vesicles? No. The packaged materials that are there, they are called as vesicles. Are they called as membranes? No, membranes are coverings. Cisternae? Yes, they are called as cisternae. They are called cisternae. So the option C is the right answer here. Next, which of the following is not a primary function of Golgi body? What is the question? We have to find out which is not a primary function of Golgi body. Is it vesicle formation? Of course, vesicle formation is a function of Golgi body. Glycoprotein formation, that is also performed by the Golgi body. Glycolipid formation is also done by the Golgi body. It is phospholipid formation. Phospholipid formation is not done by the Golgi body. So option D is the right answer here. Next question. Golgi bodies are mainly found in which type of cells? Animal cells only, plant cells only, bacteria cells only, both animals and plant cells. They are found both animals and plant cells. So option D is the right answer here. Next question. The Golgi apparatus is named after. So who is the scientist? Is it George Gregor Mendel, Camillo Golgi, George Palade, Robert Brown? So Robert Brown is the one for nucleus. Gregor Mendel is someone related to genetics. It is Camillo Golgi. In the name itself, it is their Golgi. So therefore, it is Camillo Golgi. Next question. Which organelle is closely associated with Golgi bodies? So, which organelle? Is it the nucleus? Nucleus is closely associated with the endoplasmic reticulum. Okay. So here, endoplasmic reticulum is closely associated with the Golgi bodies. Option C is the right answer here. Next, Golgi bodies are composed of multiple layers of membrane-bound sacs. The same previous question or 2 or 3 slides back I had asked a question. No? The same thing, it's in a twisted form. Golgi bodies are composed of multiple layers. So what are the multiple layers called? Is it called vesicles, lamellae, cisternae, grana? They are called as grana. Lamellae is nothing but a membrane, it's a covering. Vesicles are packaged materials and grana are usually found in, that is thylakoids, that is in chloroplast. So it has nothing to do with the Golgi bodies. The answer here is cisternae. The foldings are called as cisternae. Next question. Which part of the Golgi body is responsible for receiving proteins and lipids from the endoplasmic reticulum? So is it the Golgi apparatus? Is it the cis phase? Is it the trans phase? Or is it the cis trans phase? So what receives? So whatever protein is produced by the endoplasmic reticulum, that is received by which phase of the cisternae? So which phase? It is the cis phase. From the cis phase, they are modified and then they are sent to which trans phase? Okay, so don't get confused. First, they are received by the cis phase. So Which part of the Golgi body is responsible for receiving proteins and lipids from the endoplasmic reticulum? It is cis phase. Next, Golgi bodies play a crucial role in the modification and packaging of proteins into vesicles, ribosomes, lysosomes and peroxisomes. It is vesicles. The packaged materials are called as vesicles. Next question. Which organelle is responsible for sorting and directing proteins to their correct destination within the cell? Is it nucleus, Golgi bodies, ribosomes, endoplasmic reticulum? So it is Golgi bodies, ribosomes and endoplasmic reticulum. Ribosomes' function is to synthesize proteins. Endoplasmic reticulum's function is to deliver the proteins to the Golgi bodies, and Golgi bodies' function is to sort the proteins, pack it and deliver it to the particular sites wherever it is needed. So the answer is Golgi bodies. Next, what is the last step in the Golgi apparatus where proteins are sorted and packaged into vesicles? Where does the last step occur? So last step actually occurs in the trans phase. So the answer here is trans phase. So instead of trans phase, they can also give it as maturing phase, okay, because trans phase is also called as maturing phase. So don't get confused. So it is a trans phase. So where actually this process begins in the cis phase and it ends in the trans phase, okay. So answer is option C. Next question. Golgi bodies receive proteins from which cellular organelles? Again here, the same question, but that is, I've already asked this before, but this is in a different form. It is endoplasmic reticulum, because whatever ribosomes synthesize the proteins on the rough endoplasmic reticulum, that from that rough endoplasmic reticulum, it will move to the Golgi body. So Golgi body is the receiver of proteins from the endoplasmic reticulum. So that is option C. So this was about this session wherein we got to know about the functions and the structure of the Golgi body, and also we learned some of the possible MCQs that can be framed from this particular topic. So I hope you understood the session well. So we shall meet again in the coming session taking up a new topic under the same lesson and then learn some MCQs under it. So see you in the next session. Thank you.